intestinal protozoan that has 2 stages, trophozoite and a cyst. Which stage was the infectious stage? Cyst. Good job, you guys. And then we also talked about the trypanosoma. I love sleeping with you. So our little trypanosoma, that now the flagellum should be a little bit longer here, but the trypanosoma are also flagellates. There's a flagellum anchored here at one end. It arches back over the body of the protozoan, and then it does extend further out here. But attached to the flagellum over the central portion of the cell is an undulating membrane. We mentioned 2 different species of trypanosoma. What was the name of the species that causes African trypanosomiasis or sleeping sickness? What was that one? Trypanosoma brucei. Good. What is the vector for Trypanosoma brucei? Tsetse fly, right? And also, you guys, make sure you can recognize the scientific genus name, Glossina. Okay, so make sure you can recognize that too. And you'll remember there was a demoscope up that had peripheral blood smear with Trypanosoma brucei in it, and also a demoscope that had the Glossina tsetse fly in it. Good. There was a second species of trypanosoma that causes American trypanosomiasis or Chagas disease. What was the species name for that flagellate? Trypanosoma crucei. Good. And what was the vector for Trypanosoma crucei? The triatoma bug or reduvid bug. Good. And Professor Holland found Professor Dave Wyatt is our resident entomologist. He collected these triatomid or reduvid bugs, and so and preserve them in plastic. So make sure you guys can recognize these. This often shows up on the lab practical. So I want you to identify, provide the name, provide the protozoan pathogen that these guys carry. And let me ask you this: do you remember when these guys bite us? Are they injecting us with the Trypanosoma crucei when they bite us? No. How are we getting infected with these guys? Swiping the feces, right? Remember the feces, when they bite us, when they suck our blood, they defecate, and the Trypanosoma crucei is in the feces. So if we wipe it into the bite wound, or let's say they bite them close to our eye, if we wipe it into the eye, the conjunctiva can get infected, and then it can spread. Good job, you guys. Excellent. Okay, so those are the flagellates. So this is a little review. All right, and then we did the ciliates, and you remember the free-living ciliate that we identified, you guys? Paramecium, excellent. Right, great. They have the covered with the cilia. And then we had one pathogenic ciliate. Do you remember who that was? The Balantidium coli. Good. And just as we saw with Giardia, Balantidium coli had 2 stages. What were they called? The cyst and the trophozoite. Excellent. Which was the infectious stage? Cyst, right? And do you remember that Balantidium coli, it's often associated with warm tropical regions? And what's the non-human reservoir for Balantidium coli? Pigs. Pigs. Good. Excellent. All right. And then we moved to the amoeba. And what was our example of a free-living amoeba? Amoeba proteus, good. What's the structure of motility for amoeba proteus? Pseudopodia. Good. Pseudopodia, good. And what was the example of a parasitic amoeba that we discussed? Entamoeba histolytica, right. And remember, Entamoeba histolytica, it's an intestinal protozoal pathogen, so it too has the 2 stages, the cyst and the trophozoite. And we said that Entamoeba histolytica, it is dangerous because it can release those lysosomal enzymes on the surface of our intestinal epithelium and cause damage. It's digesting the cells of the intestinal wall, right? And so consequently it can spread. Where does it like to spread to, or where would we see a secondary site of infection with Entamoeba histolytica? Liver, right? They often will form liver abscesses. Good. And so with Entamoeba histolytica, we might see a bloody diarrhea, right? Because they're causing actual damage to the intestinal wall. Good job, you guys. Excellent. All right. So we just had a 4th group that we were going to discuss. And this 4th group is Apicomplexa. Now, many of these guys are not motile at all. I usually think of Apicomplexa as not being motile. I just read in a book there might be some that are motile, but we're going to say most of them are not motile. And the big one here is Plasmodium. And Plasmodium is such a serious pathogen worldwide. Which disease does Plasmodium cause? Malaria, that is right. And we might think, oh, malaria, it's just a tropical problem, but it's not true, you guys. The United States used to be endemic for malaria. California used to be endemic for malaria. And the reason is that we have the vector. And the vector for Plasmodium, for malaria, it's a special genus of mosquitoes. And it's always the female mosquito, and we'll describe why. There's a female Anopheles mosquitoes. And there's several different species of Anopheles. You go to different parts of the world, there's different Anopheles mosquitoes. But what's important for us to know, here in California, we have our own Anopheles mosquito. And it's called Anopheles histolytica. Named after, I think Dr. Freeborn was a UC Berkeley entomologist, and those of you that know UCD, you know about Freeborn Hall, named in his honor. So in California, we have our own malarial mosquito, and this is really important for us to remember, because if our public health system ever breaks down, if our mosquito control services ever break down, we could definitely once again become endemic for malaria, right? So as long as you have the vector, you can become endemic for that arthropod-borne pathogen. Okay, so the malaria life cycle is very complicated, and on the handout, the Protist handout, the last page has a cartoon of the malaria life cycle. So I'll just cartoon it briefly on the board, but you can just use your handout there. Okay, so with a Plasmodium life cycle, it has two distinct phases. The so-called sexual cycle, and that occurs in the mosquito, believe it or not. Okay, and then it has the asexual cycle, where it reproduces asexually, and that occurs in us, that occurs in humans. Okay, so again, I'll just kind of cartoon the major steps, again, that are on your handout. So we'll start out with our female Anopheles mosquitoes. They're often described, I love this description, as flying hypodermic syringes. Okay, so when they land on us, and take a bite, the female Anopheles mosquitoes, they have the Plasmodium parasites in their salivary glands. And what they do when they bite us, they inject some saliva into us as a little anticoagulant, because they don't want blood clotting in their little needle mouth parts. It would cause clots, and they couldn't suck up the blood. So they inject us with some saliva with some anticoagulant in it. And when they inject us with the saliva, they inject the Plasmodium. Now, I'll just give you the name of the stage, you guys, but I'm not going to ask this on the lab practical. It's just so you can kind of track some of these stages on your handout. So the form that the female mosquito injects is called a sporozoite. And what the sporozoites do is they travel via the blood to the first organ they're going to infect, the liver. And I never realized that. I always knew, well, it's probably commonly known that Plasmodium infects our red blood cells, but I didn't know it infects our liver first. So it first infects our liver cells, our hepatocytes, and it undergoes asexual reproduction. So it just splits in two, copies its genetic information, splits in two. And this will release little stages that we're going to call merozoites, this little offspring of asexual reproduction. So the merozoites then, they're released from the liver, and then they travel back into the blood, and the next cell they target are the red blood cells or erythrocytes. And this is the diagnostic stage for malaria. What you do is you take a peripheral blood sample from your patient, you make a thin slide, a thin smear on a slide, you stain it, and then you look at the red blood cells. You look at the red blood cells for little intracellular parasites. Now, they always tell us that you have to be careful, because sometimes you might have little platelets little thrombocytes. Just let me see if I can cartoon that. It's not little. Oh, good, here, okay. So let's pretend that this is a little platelet or a thrombocyte. So in a blood smear, because you have that, we're looking down in the cell, sometimes platelets will overlay a red blood cell, so it might look like you've got a little parasite in there. Okay, so folks that do malaria diagnosis, they know that they have to be careful not to mistake a thrombocyte or a platelet for a malarial parasite. And in the blood smears that you're going to look at on the demo slides, and also in your slide boxes, the blood smears are really good. You'll see a lot of the red blood cells have little purple staining dots in them. In some cases, it might almost look like a little ring. Okay, those are the malarial parasites inside. Okay, so they'll be at different stages of replicating, but those little purple dots represent the Plasmodium that have invaded the red blood cells. And again, they're going to undergo asexual replication. So they're going to divide over and over again. And then, this is really interesting, depending on the species of Plasmodium, every 48 to 72 hours, all the infected red blood cells break open at the same time. Oh, okay, yeah, it's just wild. And in fact, the physicians will ask the patients, how often do you get a fever? Every 48 hours, every 72 hours. And that helps them identify the species of Plasmodium. So every 48 to 72 hours, the infected red blood cells, they break open, they lyse, releasing, hundreds and thousands of Plasmodium that then attach to the new red blood cells and infect them. When the red blood cells lyse, it releases a lot of compounds that are potentially toxic, not only the contents of the red blood cells themselves, but also the metabolic products of the Plasmodium. And it triggers the fever that we associate with a classic malarial attack. We start shaking to try to bring up our core body temperature. And then eventually the fever subsides, but then another 48 to 72 hours, it happens all over again. So you guys can imagine that this continual ongoing lysis, it could cause anemia, lack of red blood cells. For that, the infected red blood cells, they become sticky, so they can stick to the walls of blood vessels, and they can cause clots. And this is one of the problems with one of the most serious forms of malaria caused by the most virulent Plasmodium. Okay, so I would like you guys to know this. The most virulent species of Plasmodium is called Plasmodium falciparum, or falciparum. And this malarial parasite is notorious for causing cerebral malaria. And what happens is we get blood clots forming in the brain. And again, it's because the infected red blood cells, they become sticky, they attach to the walls of the blood vessels in the brain, and you get little clots formed. And that can be deadly. So that's the most deadly of the Plasmodium species is Plasmodium falciparum. Okay, so we have anemia, we have clots that can form, we can have cerebral malaria, so we can die, obviously. Now, the groups that are most hard hit, and you're going to see the same high-risk groups over and over again, right? The very young, so children under 5 are at high risk. The very old, pregnant women get hit really hard. And of course, anybody who's immunocompromised. So in malaria endemic parts of the world where there might be high levels of HIV/AIDS, malaria can be absolutely devastating. Okay, so this is why the malaria takes such a toll on the health of humans worldwide. Now, we just want to finish our little cartoon, you guys. So we want to find out, how is it that new Anopheles mosquitoes get infected? It's really intriguing. So what happens is with some of the malarial parasites, what they'll do is they'll form what are called gametocytes. It's the equivalent of like human gametes, egg and sperm. And the Plasmodium falciparum gametocytes that you see in the peripheral blood smear, I really like them because they're so distinct. They're spindle shaped. So what happens is, again, it is in a red blood cell, but the gametocyte that you'll see in the peripheral blood smear is kind of spindle shaped. So this is the gametocyte. And this is the form that is infectious for the Anopheles mosquitoes. So if a new Anopheles mosquito comes along, and let's say I have gametocytes circulating in my blood, the female mosquito, she draws up a blood meal, she sucks up the gametocytes. Now I'll carry this on over here, you guys. So in the female Anopheles mosquito, she takes the blood meal and she ingests the gametocytes, and the gametocytes then fuse. There's fertilization. Gametocytes, they fuse, so this is a fertilization process, and then the zygote forms in the intestinal wall. Of the mosquito, and the zygote is going to grow and divide. It's going to release, grows, divides. It will produce sporozoites, and what the sporozoites do, then they migrate to the salivary gland of the mosquito. So they'll be in perfect position for the next blood meal the mosquito takes. They'll be injected into another human. Okay, so again, what we want to stress here, you guys, is asexual reproduction occurs in humans. And where is sexual reproduction occurring?
In mosquito.
In a mosquito, right? Just wild, yeah. And this is just an FYI. I wouldn't ask it on the lab practical, but parasitologists, when they talk about these parasites that use more than one host, they say the host in which sexual reproduction occurs is called the definitive host, right? So usually we think of us as being the definitive host because we want to be the important ones, right? But here it's a mosquito. You know, it's a mosquito that's actually the definitive host. Kind of interesting. Okay, now just one more tidbit, you guys. What's really intriguing is that Plasmodium is an example where a pathogen has exerted natural selection on humans. Okay, so it turns out that, remember the Plasmodium falciparum is the most deadly form of malaria. You know, if you get infected when you're a little child and you don't have proper medical help, there's a good chance you're going to die before you're 5, right? So it turns out Plasmodium falciparum has selected humans for a mutant hemoglobin allele. Okay, so a protective allele in humans. An allele is a version of a gene. Protective allele is for a mutant form of hemoglobin. It's just a single amino acid that there's a mutant form of hemoglobin. Mutant form of hemoglobin. Okay. And this mutant allele, it's called the sickle cell allele. Sickle cell allele. Okay. Now, the story is, if you're a carrier, that means you've inherited one copy of the allele from your mom or your dad, but the second copy that you've inherited from one of your parents is normal. So these would be carriers. So what they do is they have one copy of the sickle cell. We'll call it sickle cell hemoglobin. And then plus one copy of what we'll call the wild type hemoglobin. Wild type just means like the normal form, the non-mutant form. Okay, folks that are heterozygous for the sickle cell allele, meaning one normal hemoglobin copy, one sickle cell copy, they're considered carriers. And oh my gosh, there's such huge survival advantage because folks that are carriers are far more likely to survive infection with Plasmodium falciparum. Okay, so we have a much greater increased survival of folks that are carriers, increased survival in malaria endemic areas of the world. And most tropical areas worldwide are endemic for malaria. Now the heartbreak though is if both mom and dad, let's say both mom and dad are carriers, you know, and let's say they conceive a child, and the child receives a copy of the sickle cell allele from both mom and dad, then the baby's, this is trouble. Because the problem is if you're homozygous for the sickle cell allele, in the old days, before there have been advances in medicine, a lot of these kids often wouldn't survive to adulthood. Okay, so in the old days, before we had some medical advances, many of these children died. Many of these children died, and the reason is, what happens under low oxygen concentrations, the sickle cell hemoglobin, this is wild, you guys. It's almost like the prion story. It misfolds because it has this one wrong amino acid in it. It misfolds. And when it misfolds, it tends to stack. Okay, now, if we look at the consequences of that, normal red blood cells, you know, they're oval, they're flexible, right? So they can squeeze through our tiny, tiny capillaries, right? So this would be a regular red blood cell. It's flexible. It can squeeze through these little tiny capillaries. But what happens is if we have a sickle cell crisis, okay, when the mutant hemoglobin, when it misfolds, it stacks. And it can cause these long filaments that causes distortion of the red blood cells. Okay, so these little lines are just supposed to represent the stacked hemoglobin. And it will cause the red blood cells to distort in shape. And one of the shapes that you'll see in during a sickle cell crisis, some of the red blood cells, they look like the metal sickles they used to use in the old days to harvest grain in the fields. And that's why it's called sickle cell. The problem is these guys are no longer flexible. So when the cells are sickling, they can't squeeze through the capillaries. They create little clots, right? And they're gonna block off flow of blood to vital organs. Okay, so this is why in the old days, kids that had two of the sickle cell alleles, a lot of them never made it to young adulthood. It was, you know, heartbreaking. Now with medical advances, the kids that have two copies of the sickle cell allele, they can definitely make it to adulthood. You know, things are looking much better, but it's still really tough when they have one of the sickling crises. It's really very, very hard. Now, the question is, you know, so what the heck's going on here? Why should one copy of this allele, you know, be protected? Well, first of all, if you're a carrier, you don't have sickle cell crises, right? Half your hemoglobin is okay, so you don't end up with the sickle cell crises like kids that have two copies. And what I've seen so many explanations for why the one copy helps. And the last one I saw that I kind of liked is one idea was if you're a carrier of the sickle cell trait, you know, half your hemoglobin's normal, half of it's sickle cell. If the Plasmodium invades your cells, the Plasmodium will cause a drop in O2, and you'll get some partial sickling. You'll get some partial sickling of like half your hemoglobin, right? And that's a marker for your spleen to destroy those red blood cells. Your spleen is an organ that helps to destroy, recycle damaged or old red blood cells. So we'll get maybe slight sickling, not significant. Slight sickling. And I'm going to put a question mark for you guys because you'll read all different kinds of mechanisms for why they think this helps. But it gets slight sickling, and that will cause rapid destruction by the spleen. Some papers say that with Plasmodium invasion, you get a little bit of sickling, and these red blood cells will just break more easily. That's another explanation. Rapid destruction, we'll say by spleen. And what happens then is this prevents the Plasmodium from replicating in high numbers. If as soon as the Plasmodium invades a red blood cell, they're destroyed, they can't replicate. And so consequently, they can't spread to other cells. So this might be one possible explanation for why the carrier state for sickle cell is protective against the most virulent forms of Plasmodium. Right? Okay. And again, so this is often used as an example where a pathogen has actually exerted natural selection on human populations. Okay, so many people whose ancestors lived in tropical areas of the world where malaria was endemic, those populations are enriched for the sickle cell allele because it was a huge survival advantage, right? Okay, but one of your colleagues a few years ago